Hello, long hauler friends. I came across something today that I thought that you guys might find interesting. This is Dr. Danielle Beckman. She's a neuroscientist and researcher at the California Primate Center. She does a lot of work with UC Davis, and a lot of the information that she shares has a lot to do with high resolution microscopy. And today that she posts that it's Fluorescence Friday, and wanted to include the first photo she took of the microglia cells fusing with each other. The one that initially sharing was with HIV. Because as we know, the HIV virus can cause microglial cell fusing. Well, she goes on to say that she's looked at brains under the microscope for many years now, but knowing that neurons and glial cells can fuse with each other wasn't something she'd ever learned, because most viruses don't even directly cause damage to the brain. When they do, it's because they killed their host cell. However, some chronic neurological sequelae of viral infections cannot be explained only by neural cell death. It's known that in non-neural tissues, so like non-brain tissues, non-central nervous system tissues, that viruses fuse with host membranes to enter the cell. Then at that point, it hijacks the cellular machinery to produce viral components and sometimes confers the ability to fuse with neighboring cells. And ultimately, the results in a formation of multinucleated cells. Now, this is a very rudimentary drawing that I did, but you can see here that as the cells fuse together, it makes it appear like there's multinucleated cells. So then you can imagine that, picture these cells that would have virus within one of them. What would happen is it allows the virus to propagate from cell to cell without having to leave the cell. Essentially infect other cells. It's able to do so because it's fusing them together. Now, one important implication regarding why it's important that the viron wouldn't want to leave the cell is because our antibodies live outside of the cell. And more rudimentary drawings, but here I just use some simple cells. The virus would be in green, and that's after it's already infected. This is infection, once it has made it inside a cell. So if it would have to leave the cell to infect another cell, that's where the virus faces a little bit of a threat. So our antibodies, pictured in yellow there, live outside of the cell, and there we see the virus leaving each of the cells so that it could go maybe infect a new one. But with the antibodies outside, they could tag and destroy. Like seen here. Much better picture. And one of the places that we would really see this occur is in RSV, or respiratory syncytial virus, where it fuses lung cells together. But guess what? Viral-mediated fusion of the brain cells also exists, but it's poorly understood. Irreversible fusion of brain cells causes a disruption in neuronal communication and reveals another possible mechanism to explain the chronic neuronal malfunction, even many years after the initial infection. Now, here's a picture of a neuron, and we can see here that there is even a direction of an impulse, and which way the pulse will flow. And when you get groups of them together, like pictured here, the impulse or the signal really becomes almost like a game of telephone, where they're passing a signal or a message on to reach certain parts of the brain. Now, say for example, you put your hand on a stove. You will have pain receptors that, once activated, start sending that impulse or signal in a telephone-like fashion to the brain, and then the brain signals move your hand back down the line in another telephone-like fashion to tell the muscles to move. We now know that SARS-CoV-2 can also cause brain cells to fuse, especially inducing neuron-to-neuron fusion. So instead of firing independently to propagate signals throughout the brain, like a game of telephone where one, would tell the other, would tell the next one in line and the next one, the fused neurons fire simultaneously. You think of the firing, that's the message that's being sent. It winds up being several of them that are now fused together saying that message at once. This massive amount of synchronous activity is similar to seizure-like events, and I thought that was really interesting. The desynchronized neuronal activity caused by fused neurons can also help explain symptoms like brain fog. And when these events occur in the autonomic nervous system, for example, it could help explain the origins of dysautonomia, which we often see in long CE patients. Anyway, here's the photo that she had included for Fluorescence Friday of the microglial cells of the brain fusing together, seen in this specific instance with the HIV virus. And then she included these articles in her post as well. Unfortunately, as she stated, this fusion is irreversible at this time. But identifying that this could be a potential cause could potentially lead to developments in the future for how we could address this issue.